Hi, my name is Ethan Smith, and today I'll be talking about our project related to the inference of long non-coding RNA regulatory functions from pseudo-time ordered single-cell data. So first I'd like to start off by talking about what long non-coding RNAs are. Long non-coding RNAs are defined as RNA molecules longer than 200 nucleotides, which are not translated into protein. They are often involved in gene regulation and from that derive important functional roles in many important processes such as development and differentiation, cell cycle and apoptosis, immune response, and oncogenesis, just to name a few. Long non-coding RNAs have a few unique features that also make them quite challenging to study. The first step in studying long non-coding RNAs is to accurately identify and annotate them within the genome. However, this step proves to be more challenging than it may initially first sound. First, long non-coding RNAs are often lowly expressed and often display tissue or cell type specific expression patterns, making them difficult to identify from traditional bulk RNA-seq data. In many cases, background noise from alignment errors or spillage from cell-free ambient RNA make manual curation of long non-coding RNAs practically impossible. Additionally, long non-coding RNAs typically have very low sequence conservation, preventing the use of gene prediction algorithms, another common method of gene annotation. Secondly, the functions of long non-coding RNAs are not typically defined by their sequences, as is common with protein coding genes. Instead, they are typically determined by the long non-coding RNA's secondary or tertiary structures and by interactions with other biological molecules. As a result, predicting the functions of long non-coding RNAs through an in silico manner is actually quite difficult. To approach this problem, we decided to leverage the enhanced resolution afforded by single-cell RNA sequencing. Single-cell RNA sequencing allows us to address many of these problems by performing transcriptomic profiling at the individual cell level. This allows us to capture lowly expressed, highly cell-type specific long non-coding RNAs in a manner that would not have been possible through bulk RNA sequencing. Another great benefit of single-cell RNA sequencing is that having this resolution at the individual cell level also allows us to capture the inherent heterogeneity that is present among the different cells within our samples. Using this heterogeneity, we are also able to order the cells based on their progression through distinct biological processes, providing us with a sort of developmental or pseudo-time ordering through which we can track the expression of any gene we would like. For instance, by looking at the expression patterns along our pseudo-time trajectory, we can identify instances where a specific long non-coding RNA is consistently expressed before a particular marker gene associated with our biological process of interest. This analysis enables us to infer regulatory relationships between the long non-coding RNA and the marker gene related to the process we are studying. This is only possible because the cells from our data have been ordered based on their progression through our given process of interest. Now at this point, you may be wondering what evidence is there that these long non-coding RNAs are actually poorly annotated, especially within genomes of commonly studied organisms. To answer this question, we decided to look at a rhesus macaque single-cell RNA-seq dataset from an ongoing dengue-Zika cross-immunity study. In this dataset, we searched for regions of the genome with transcriptional activity that lacked annotation in the current rhesus macaque reference. To accomplish this, we used a pipeline similar to the one described by Wang et al., referenced in our proposed workflow. For our purposes, we refer to these regions as unannotated transcriptionally active regions, or UTARs. As we can see in Figure 1b, when plotting the expression of these UTARs against the UMAP embeddings generated by previously annotated genes, shown in Figure 1a, several UTARs display cell type specific expression, which is indicative of their potential biological relevance. Additionally, in Figure 2, we observed that when we regenerated these UMAP embeddings using only the UTAR expression information, the cells in our dataset cluster by cell type, suggesting that these UTARs do indeed contain biologically relevant information. In total, over 80,000 UTARs were identified in this one sample of just over 10,000 cells, suggesting that there are many long non-coding RNA features missing from the current rhesus macaque reference annotation. Given this preliminary evidence, it is clear that a more in-depth pipeline for the annotation of these long non-coding RNA features is needed. However, several additional filtering and refinement steps will be needed to ensure that these UTARs we have obtained are indeed long non-coding RNA features. As outlined in our proposed workflow, we plan to filter these UTARs to exclude things such as unannotated exons of known protein coding genes. Additionally, we plan to develop a metric to score each of these UTARs based on their likelihood of being long non-coding RNAs. Once this is complete, we will combine our newly produced long non-coding RNA annotations with the previously annotated long non-coding RNAs from the current reference annotation. This combined list of long non-coding RNA features will be what we use in step 3 of our workflow to infer potential long non-coding RNA marker gene regulatory relationships. The next step of our proposed workflow involves performing the trajectory inference to obtain our pseudo-time ordering of cells. To accomplish this, we plan to use a well-established method by Kamimoto et al. That involves construction of cell-type-specific gene regulatory networks from a combination of our single-cell RNA-seq data and a public single-cell attack-seq reference. Once our cells have been ordered, we can then identify these sequential patterns of long non-coding RNA marker gene expression and use a time series analysis method, such as Granger causality, to help us determine if a regulatory relationship is truly present. The underlying benefit to this proposed workflow is its generalizability. While in our case, we are applying it to rhesus macaques to study long non-coding RNAs in immune systems, this method is species agnostic and can be applied to study long non-coding RNAs in virtually any single-cell RNA-seq dataset in principle. Thanks for listening.